So if you are watching this, it's because we didn't have class because of the snow on Thursday. So what I'm going to do is, and this is Dr. Merkhofer, they'll be listening to me right now. So what we're going to do is we're going to go and we're going to continue on with what we did in the last class. Now, the first thing you're going to need to do is open your DNA Master program. So you can see I've on the Windows desktop, and when I go through these some of these steps, it's going to look different depending on the computer you're using, but you'll be able to hopefully find these different things. Also, keep in mind, you can pause this at any time. So if you feel like I'm going too fast, just pause it. You can rewind it a little bit. And you can always send me or Dr. Bala an email at any time, and we'll answer any questions you have. So we're going to open up our DNA Master program, and this is either going to be on your desktop or through the VPN, the link that I sent by email. So I'm going to go ahead and open DNA Master. And we're going to load, so last class we uploaded our FASTA file and we converted that into a DNA Master file. Some people weren't able to save it, so I'm going to go through that step again. So the genome file, all those ATCs and Gs, is on your eClass page. So if you go to the eClass page for the class and go to DNA Master Resources, there's going to be a file that says Octavius Rex FASTA file, this one here. I'm going to download it. And what I'm going to do is I'm going to make sure that I know where that is. I'm going to open that, and I'm just going to move that to my desktop. And the reason I'm doing that is it's going to be easy for me to find when I'm trying to load it. So I just downloaded the Octavius Rex genome sequence file, and I'm going to go back to DNA Master here, and I'm going to open it. I'm going to begin a new blank project. I'm going to click Begin. Then I'm going to go to File, Open, and I'm going to choose FASTA multiple sequence file. And while we're going along, I'll try to indicate where this is in the annotation guide as we're doing this. So what we're doing right now is on page 23 in the annotation guide. If you left your annotation guide at home, or you don't have it with you, the annotation guide is on eClass as well. So I'm going to click FASTA multiple sequence file. It's going to take a minute to load, depending on how fast your computer is. And I'm going to go to desktop. I'm going to look for that file that I just downloaded, which is the Octavius Rex FASTA file, and I'm going to open it. And what you're going to see here is that the sequence got imported into our program here. So these are the 41,880 ATCs and Gs for our genome. So we need to convert this into a DNA Master file. So to do this, I need to resize these windows a little bit. So I'm going to make this window a little smaller, like this, so I can bring it up to move this up. And I'm going to, because I need to see this export button here. So if you can't see it, it's because your window is too far down. Just move it up like that. I'm going to click on Export, and then I'm going to click on Create Sequence from this entry only. And what that's going to do is that's going to convert this into a DNA Master file. What I'm then going to want to do at that point is I'm going to want to save this file as a DNA Master file. The more often you save a file, the better, because if something happens, you're going to want to go back to the previous file. So I'm going to go to File, plugged in my jump drive. I'm going to go to File. I'm going to save as DNA M5 file, And I'm just going to save this onto my jump drive. And this is, I'm going to leave this as extracted from FASTA library Octavius Rex. And save. So now what I've done is I've converted this into a DNA master file, and if I wanted to reopen this, so I can say I was to close out of the program here, and I was to reopen DNA master, I should be able to open it from this location. So open a previous DNA master project. And extracted from FASTA library Octavius Rex. And it'll bring me right back there. So this is just to check to make sure that you can save the file and open it back up. So the next thing that we're going to want to do, so this is on page 25, I just want to show you what some of these different tabs are here. This is the main window of DNA Master that we're going to use. The overview is going to give you information. There's not much in there right now because we haven't done a whole lot with our genome. So actually, before we will look at this in a minute. The next step is going to be to perform what's called the auto annotation of this genome. So what do we mean when we say annotation? So we can break annotation into two parts, what we call structural annotation and functional annotation. And we're going to be doing both of these. So the structural annotation is to identify the parts of the genes that are important for delineating them from other genes. So finding what are called open reading frames, and we'll talk about open reading frames in just a minute. Gene structure, so where are the promoter regions? Coding regions, so the coding regions are the parts of the genes that make the proteins. And regulatory motifs, like I said, promoter regions and some other things that we'll talk about later. But also there's what's called functional annotation, which is determining what those genes do. So figure out what their function is in the cell. So for each of the genes in our genome, we're going to try to determine not only where the borders are of the gene, so where is the start site and the stop site of the gene, but also what is the function of the gene. So if we go back to DNA Master here, we're going to do what's called an auto annotation of this genome. So on page 27, you can see we're going to click on Genome and Auto Annotate. And it's going to give us a window here. And on page 28 of your annotation guide, it's going to show you the default or the settings that you should be using for your auto annotation. So you're going to want to make sure that the boxes look just like they do on page 28 in figure 4.2. So this is the first tab, the gene calling tab. Make sure everything looks the same. Tags and comments, make sure everything looks the same there. And you may have to change these. So for example, your start may be 10 and your spacing may be 10, but make sure that they are 1. So we're looking at page 28 and making sure each of these tabs matches up with the settings that are in there. I've already run this on this auto annotation on this program before, so mine are already set. You're going to have to check on page 28. So once we have that all done, we're going to click on the Annotate button. And it'll ask you if you want to erase features and annotate genome. We'll say yes. And what it's doing is there are programs out there that will predict where genes are simply based upon what the sequence is of our genome. And it is running these programs, and you should see this auto annotation log come up. It's running these programs to predict where the genes are. Now, it does a relatively good job with this, but it's never going to be completely correct, and that's why we still have to annotate this by hand. So what this is showing me here is that it ran the auto annotation of this file extracted from FASTA library Octavius Rex. So I'm getting a text right now. MSMC will be closed on Thursday until 5 p.m. So you will most definitely be watching this tomorrow. Excellent. So the auto annotation has been done. It has predicted our genes. And now what we're going to do is we're going to take a look at it. Before we actually start analyzing this, the first thing that we're going to want to do is we're going to want to save it, because we've done the auto annotation. So I'm going to go to Save As. And we might have to get rid of that window first. There we go. We'll go to Save As, and again I'm going to go to my C drive, or I'm sorry, my jump drive. And here, it's not a FASTA file anymore. Now you can name it whatever you want. So this is our Octavius Rex. We've started to annotate it. So I'm going to say Octavius Rex annotated 2.8, because that's today's date. So that way I know that this is the most recent version of this. And if it says compression error, don't worry about that. I have no idea what that means. So we've got our annotated genome. So what I'm going to do is I'm just going to make this a little smaller. So you're going to see a few things here. First, we have the Overview tab. So this is going to give us any information. And there's not going to be a lot in here. You have your length. The number of features is the number of genes that were predicted. And that kind of correlates with the number of open reading frames. So we'll talk about that in just a second. If you look at the very bottom of this, this is a map of where it predicted the different genes. So each green shape here is a gene on one of the strands of DNA. The red ones are genes on the other strand of DNA. And we're going to annotate each and every one of these. If you click on the Features tab, what you'll want to do is right-click on this box here. On a Mac, if you have a mouse, you can right-click. You also, if you're using your mouse might not let you right-click, or if you're using a trackpad, you might have to do Control-Shift-Click. And you're going to click on Widen Feature List, and that's going to show us some important information for each of these predicted genes. So, like I said, it predicted 61 genes. So you can see all 61 here. And for each one, it gives us the coordinates of what it predicted as the start site, which is the 5' end here. The stop site, so the translational stop site, which is the 3' end, and the length of each of these genes. We'll talk about what this information over here means in a little bit. But it gives us information for each of these genes here. There's a References tab. You won't need to use this at all. The sequence, this is showing you the entire sequence of the genome. You generally won't use this either. And the last is the Documentation tab. And the Documentation tab is something that we will use periodically. What the Documentation tab does is it keeps track of everything that is done in DNA Master. So this is kind of another backup. If something happens where DNA Master crashes, you may at some point need to take this information in the documentation tab and import it back into another part of DNA Master. And we'll talk about that as we go along. But mainly, we're going to be working in the overview and the features part of the DNA Master. So, one thing that is in here, it does say the open reading frames, or the number of ORFs. ORFs stand for open reading frames. So let's talk about what open reading frames are. So let me go to my PowerPoint here. So we talked before how there are 6 possible reading frames in a DNA sequence, because codons are in sets of 3 and the genes can be on either strand. So here we have 6 different open reading frames, or I'm sorry, 6 different frames for our 2 DNA strands. And you can see it's broken up into kind of colors here. And you'll notice some important things. You see these ATGs. So ATG is the most commonly used start codon in this bacteria. You can also have GTG and TTG less often as a start codon. So every gene that we identify from our virus is going to start with one of these 3 codons. So this is a signal to start making a protein. Every gene that we annotate from this virus is also going to stop with one of these 3 stop codons, TAA, TAG, and TGA. These are used about equally. So you can see here these ATGs are circled. And we also have the stop codons that are circled. Anything between a start codon and a stop codon is considered an open reading frame. And this isn't a very good example because here we have a start codon and we don't have anything past it, so there might be a stop codon down here. Here we have an ATG, but then a stop codon right after that. So we actually don't have any open reading frames here. So between 2 stop codons is not an open reading frame. An open reading frame is the distance, the sequence between and including a start codon and a stop codon. Those are where potential genes are found. Not all open reading frames are genes, but all open reading frames could potentially be genes. So another way of kind of looking at this is we look for potential coding regions, potential places where genes could be, by looking for these open reading frames. So if this is a start codon here, ATG, and this is a stop codon, this entire part of the sequence is an open reading frame. And depending upon what that sequence is, we may decide that that is a gene. And there are different things that we're going to look at to determine if in fact these are genes or not. So again, an open reading frame is the distance, is the genomic area between a start codon and a stop codon in any of the 6 reading frames. So why is this important? Well, when we look at our sequence, we're going to want to look for these open reading frames. And the way that we can do this, and so also just kind of going back to the annotation guide, we talked about page 30 and 31, viewing the documentation, viewing the feature table. I'd recommend reading through that. We're going to go to page, so on page 33 it talked about viewing the sequence in the sequence tab, and we looked at that. So this was the sequence in the sequence tab right here. But we're going to go to page 34 right now in the annotation guide, which is viewing open reading frames in the frames window. So this is something that we're going to do pretty extensively. Now, the pictures that they use in your annotation guide are for a different virus. So don't expect the pictures in the annotation guide to match up with what you see with your particular sequence. But they should look similar in a way. So we're going to go to DNA, and we're going to click on frames. And what this is showing is this is a graphical representation of the 6 reading frames for the virus that we're studying. There are a bunch of lines here of 2 different sizes. Anything that is a line that goes from the top to the bottom of one of these rows, like this here, like this here, like all these here, these are stop codons. So in this frame, this is a stop codon, this is a stop codon, this is a stop codon, this is a stop codon. In this frame, these are stop codons, these are stop codons, these here are stop codons. You also have these shorter ones. So I have a whole bunch right here. These are potential start codons. So these are ATGs, TTGs, GTGs. These are sequences that could potentially be the beginning of an open reading frame. So I'm going to zoom in a bit here and use an example. So if we look at, say, this frame right here, from this start codon all the way down to this stop codon is an open reading frame. From this start codon to this stop codon is also an open reading frame. It's a smaller open reading frame. From this start codon to this stop codon, this is an open reading frame. It's a smaller one. There will be large parts of the genome in a certain frame that don't have an open reading frame. There's no open reading frame here because there's a stop codon, and then there's not another start codon until the next stop codon. So if we look, for example, right here, this start codon to this stop codon is an open reading frame. The top 3 rows are one strand of DNA. These go from left to right. The bottom 3 are the other strand of DNA. These that go from right to left. So if we're looking for open reading frames here, we would go, say, from this potential start codon over to this stop codon. Or down here, this potential start codon to this stop codon. I'm going to zoom out and to about this level here. And we can ask the program to tell us which open reading frames it has predicted are genes. To do that, we click on this button over here that says ORFs, ORFs. And we're going to click on that, and it's going to put in these green and red boxes. And in this area, there's only green boxes. What this is showing is which open reading frames DNA Master thought were genes. So we can zoom in on the plus magnifying glass here if we want. And you can see, for example, this open reading frame from this start codon, number 13, to this stop codon, DNA Master thinks is a gene. And then a little further down the DNA, and this is in a different frame, this start codon to this stop codon, it thinks is a gene. And then right after that, but again back in frame 2, from this start codon to this stop codon, it thinks is a gene. So these are the predicted. It doesn't mean that they're right, it means that this is what is predicted. And if you ever want to select on any of these, so let's say we want to select number 10 here, you can click anywhere to the left of the stop codon, and it's going to select the open reading frame over to the start codon that you want, just like that. And then if you want to remove those, you click this back green arrow. So let's say I picked this one. All right, I'm done with that. I can just clear it out, just like that. On page 36, and the selecting that I just did is on page 35. On 36, they talk about this RBS. We will come back to this. We're not going to talk about that quite yet. So I'm going to click back on this window right here. So just to recap what we've done so far, we imported our FASTA sequence into DNA Master. We saved it as a DNA Master file. Then we auto-annotated our genome, which means that the DNA Master picked what it thought was the best genes. And then we looked at different ways of viewing this, including the frames window here, which you will end up using quite a bit. So the next step is going to be doing another BLAST search. So the BLAST search that we did previously was we blasted the entire genome of Octavius rex against all of the genomes in the phages database. And that gave us an idea of which of the phages were most similar genetically to our phage. But what we also want to do is we want to take all of these potential genes and blast them individually. And what that means is we have 61 different sequences here that DNA Master thinks is a gene. We're going to ask DNA Master to compare each of those sequences to the sequence database and tell us, does this match up against any known genes? And this will help in our annotation process. So in order to do this, we go to genome and blast all genes. Now, the instructions to do this are on page 37, but don't do it. The reason I'm asking you not to do it is this takes a long time. It will take hours and hours to run. If you start blasting this, you will not finish for hours, and you won't be able to run anything else in your program. So do not blast it. I'm just going to walk through what the instructions say here. So what this is telling it to do is it's saying take all of these genes and compare it against, so blast all these genes and compare it against the database to tell us what each of these potential genes, each of these 61 potential genes, matches up to. Like I said, we're not going to do this. We have already done this for you. So if you've gotten to this point, what you can do is you can X out of this, and you can actually X out of the window here. And you can save this before closing, because this is our extracted, or I'm sorry, we can save this as our annotated. So this can be our Octavius rex annotated 2.8, and it's telling me my jump drive is full. But you can save it on your jump drive, and then once you save it, then you can close it out.